Before we get into mutations and how they affect a person's DNA, let's review the basic functions of DNA. First, it stores genetic information that will determine the characteristics and functions of an organism. Next, DNA can go through replication, which is a wonderful process where it can actually make exact copies of itself, which is essential for cell division and growth. DNA also is involved with holding the directions on how to make a protein, so it has two more processes as well: transcription and translation. DNA is transcribed into RNA and then translated into proteins. Finally, DNA is responsible for passing genetic traits from parent to offspring. So then what happens if the process of DNA replication does not go as planned? Insert mutations, a change in the sequence of bases in DNA or RNA. Mutations result in a change in the genetic code. Everyone has mutations. In fact, most people have dozens or even hundreds of mutations in their DNA. Mutations are essential for evolution to occur. They are the ultimate source of all new genetic material, new alleles in a species. Although most mutations have no effect on the organisms in which they occur, some mutations are beneficial. Even harmful mutations rarely cause drastic changes in organisms. Up until now, we've thought of the nucleus containing the DNA in little floating strands. In reality, it's a little bit more complicated than that. Because we are eukaryotic cells, we have a nucleus, and yes, it contains the DNA, but the DNA is actually packaged very neatly in a super-organized bundle called a chromosome. Each and every one of the cells in your body contains 23 pairs of chromosomes, half of these that you received from your biological mother, and the other half of the chromosomes coming from your biological father. Within the chromosomes is the DNA, and if you zoom in on a portion of the DNA strand, you would then find the alphabet sequence of adenine, thymine, guanine, and cytosine. Sometimes chromosomal mutations occur. Chromosomal mutations refer to alterations in the structure or number of chromosomes in an organism's cells. These mutations can have significant impacts on an organism's development and function. A chromosomal mutations will change the entirety of the chromosome structure. Within chromosomal mutations, we have a few different types depending on what happens. The first type is called inversion. Inversion occurs when a segment of the chromosome breaks off and reattaches itself to the chromosome, but in the reverse orientation. This means that the base pairs of the DNA within that segment are flipped or reversed. While the genes themselves remain intact, their arrangement is reversed. Next is translocation. Translocation happens when a segment of one chromosome breaks off and attaches to another chromosome. This results in the transfer of genetic material from one chromosome to another. Duplication involves the presence of extra copies of a segment of DNA within a chromosome. This can occur due to errors during DNA replication or recombination processes. Duplication can lead to an increase in the number of certain genes, potentially resulting in genetic disorders. Deletion. Deletion occurs when a segment of the chromosome is lost or deleted during cell division. This results in the loss of genetic material from the affected chromosome. Deletions can vary in size, from small segments to entire chromosomes. Depending on the genes involved, deletions can have varying effects, ranging from mild to severe consequences. Each of these types of chromosomal mutations can have diverse effects on an organism depending on the specific genes and regulatory elements involved, as well as the extent of the structural alteration. Mutations can contribute to genetic diversity within a population and can also be associated with genetic disorders. We will talk more in depth about genetic disorders next unit. Point mutations are small-scale changes in the DNA sequence that involve the alteration of a single nucleotide base pair. These mutations can occur spontaneously during DNA replication or as a result of exposure to mutagenic agents such as radiation or certain chemicals. Point mutations can have various effects on the genetic code, depending on where they occur and the specific change that takes place. There are several types of point mutations. The first we will cover is substitution mutations, which typically involve the replacement of one nucleotide base with another. There are three possible outcomes of substitution mutations: silent, missense, or nonsense. Silent mutation. In some cases, a substitution mutation does not result in any change in the amino acid sequence of the protein encoded by the gene. This occurs when the substituted nucleotide still codes for the same amino acid due to the redundancy of the genetic code. Missense mutation. A missense mutation occurs when the substitution results in the coding of a different amino acid. This can lead to changes in the protein's structure or function, which may have various effects on the organism. Nonsense mutation. A nonsense mutation occurs when the substitution creates a premature stop codon in the mRNA sequence. This leads to the premature termination of protein synthesis, resulting in a truncated, non-functional protein. Here's a good example of an original DNA sequence and then the mutated sequence. See how there is one single change in a nucleotide? That one base pair change is how we know it is a point mutation. The next type of point mutations are more specifically named frameshift mutations, because when a nucleotide is added or deleted from a sequence, the entire reading frame of a codon is changed for the rest of the sequence. Deletions remove nucleotides, and insertions add nucleotides. More specifically, insertion mutations involve the addition of one or more nucleotide bases into the DNA sequence. This can disrupt the reading frame of the genetic code, leading to significant changes in the amino acid sequence of the resulting protein. Insertions can cause frameshift mutations, where the reading frame of the mRNA sequence is shifted, often resulting in the production of a non-functional protein. Deletion. Deletion mutations involve the removal of one or more nucleotide bases from the DNA sequence. Like insertions, deletions can also cause frameshift mutations if the number of deleted bases is not a multiple of 3, leading to a shift in the reading frame of the mRNA sequence and potentially producing a non-functional protein. A frameshift mutation is more damaging than a silent, missense, or nonsense point mutation because by adding or deleting a nucleotide, the entire sequence that follows the mutation is affected.